Hello dear friends, welcome to a new video of annotating first aid. Now we are going to look on to pathology-cellular injury on page number 204. Okay, this is what I studied today and let's annotate together. Okay, and from this video onwards we won't be reading each sentence cover to cover because we don't have enough time for that. I'm just telling you what are the things that I have annotated from the recent PYQs and of aims and recent NEET questions. Okay. Skeletal and heart muscle, there is only hypertrophy, no hyperplasia. That is a thing that is commonly asked. Skeletal and heart muscle, only hypertrophy, there is no hyperplasia. Question will be like, which of the following won't undergo hyperplasia? It is skeletal and heart muscle, they commonly undergo hypertrophy only. So in NEET, it was asked number of base pairs in the human DNA is 3.2 billion. 3.2 billion number of base pairs are present in the human DNA and they are distributed in 23 chromosomes. Then non-coding regulatory RNAs. There are some non-coding regulatory RNAs in our body. They are useful for the regulation of gene expression. It is useful for the regulation of gene expression. Non-coding regulatory RNAs are actually functional, but they are non-protein coding. Even though RNA is produced, they are not translated to protein. They are non-protein coding areas of DNA that are transcribed to RNA but never translated to protein. Examples are microRNA, miRNA and LNC RNA, long non-coding RNA. They cause regulation of gene expression. Regulation of gene expression is caused by that. miRNA is formed post-transcriptionally, but they are not translated to any protein. Okay. Now we go on to a recent NEET aims question. Anaplasia is a lack of differentiation and is an indicator of malignancy. Then there was another question in NEET. In muscle hypertrophy, the alpha isoform of myosin heavy chain is replaced by the beta isoform. Alpha isoform of myosin heavy chain is replaced by the beta isoform. Why is this happening? During muscle hypertrophy, the muscle is doing a smart work, that it is trying to conserve the energy for contraction. In order to have an energy economic contraction, slower contraction, the muscle converts the alpha isoform to beta isoform. Alpha isoform is the adult kind of isoform, beta isoform is the fetal isoform. Next we shall go into another NEET question. Thrombosis is initiated by endothelial injury, and we also know that endothelial injury is a part of our Virchow triad. Endothelial injury, stasis or turbulent blood flow and hypercoagulability. Then arterial and venous thrombus, just an extra edge. Arterial thrombus is formed inside the arteries, and since there is high velocity blood flow occurring in the arteries, what would be the reason for a thrombus to form? It is endothelial injury that caused an arterial thrombus, and the arterial thrombi is white in color. Venous thrombi occur due to stasis of blood inside the veins, and it is red in color. Okay. Then another thing, we know that in dysplasia, there are many changes, out of which one change is increased nuclei is to cytoplasmic ratio. So we need to know what is a normal nuclear cytoplasmic ratio. That is 1:4. If one part of nucleus is there, 4 parts of cytoplasm will be there. But that is converted to 1:1 in malignancies. 1 part of nucleus, 1 part of cytoplasm, like that. So another NEET question is name a connective tissue metaplasia, or find out the connective tissue metaplasia from the following options. The answer can be myositis ossificans. In myositis ossificans, inside the muscle, bone is getting formed. So my muscle is getting ossified to bone due to some after hemorrhage and all. After hemorrhage into the muscle and all, muscle is getting ossified to bone. That is a connective tissue metaplasia. That was a recent NEET question. Then a commonly asked question is find out which of the following are not going to undergo hyperplasia. So the answer most commonly would be skeletal muscle and cardiac muscle. Skeletal muscle and cardiac muscle, both of them, they normally undergo hypertrophy only and not hyperplasia. Okay, so we have got some extra edge points also. Most common cause of cell injury is hypoxia. Most sensitive tissue to cell injury or hypoxia, most sensitive tissue to hypoxia is our neuronal tissue. Then another extra edge, whenever there is cardiac muscle hypertrophy, there are some transcriptional factors that are involved here. Cardiac muscle hypertrophy, the transcription factors involved are GATA4, MEF2, NFAT. GATA for MEF to NFAT. There are many A's and T's, GAT, FAT, and then somehow you remember MEF2. GATA for NFAT and MEF2 are the cardiac muscle hypertrophy transcription factors. Then recent AIMS question. The page number is 205. You are going to annotate it here. Here we have got cell injury, reversible cell injury and irreversible cell injury. The steps are very, very important because they are very frequently asked. They give the steps of reversible cell injury and ask us to find out which among the following is an irreversible cell injury kind of a step. And in AIMS it is asked, most important light microscopic finding of irreversible cell injury is nuclear changes like pycnosis and karyolysis. Nuclear changes like pycnosis and karyolysis is the most important irreversible cell injury light microscopic finding. Okay, then we have got another question. Like I said, they would list in the options some points from this reversible cell injury and ask us to find out which among them is an irreversible cell injury kind of finding. And in one question it was amorphous densities or inclusions in the mitochondria. That was the point from the irreversible cell injury. Okay, then metaplasia in the lung can occur due to deficiency of vitamin A. We know that in lungs there can be metaplasia, and metaplasia in the lung, that is squamous, is occurring due to deficiency of vitamin A. So what are the different types of metaplasia that we know? In airway, there is normal pseudostratified epithelium is getting converted to abnormal squamous epithelium in smokers and vitamin A deficient people. Okay, pseudostratified epithelium in the airway is converted to squamous epithelium in smokers and vitamin A deficient people. Then in urinary bladder, we know that the normal epithelium is transitional epithelium with umbrella shaped cells, but it is getting converted to squamous cells due to urine bladder stones. Transitional to squamous. In airway it is pseudostratified to squamous. In esophagus it is different. Esophagus is an opening kind of means esophagus, the tube is in contact with the external environment through mouth. So it is actually the squamous epithelium that the esophagus normally has. It is getting converted to columnar epithelium of the gastric columnar epithelium, and this metaplasia has got a name called as Barrett's esophagus. Barrett's esophagus proves a risk factor for adenocarcinoma. So squamous to columnar metaplasia in esophagus occurs in chronic GERD. Then cervix, in cervix we have got glandular epithelium. There are lots of glands in cervix. We have got glandular epithelium that will convert it to squamous epithelium. Squamous epithelium in patients with low pH in the cervix. In patients who have got a very low pH of cervix, glandular is converted to squamous. In muscle, we already know the metaplasia happening is the normal muscle tissue is converted to bone called as myositis ossificans. It usually occurs after some kind of hemorrhage. In NEET, there was a question. Most common metaplasia is columnar to squamous. We know that the squamous metaplasia is the most common metaplasia by now. Airway, it was pseudostratified to squamous. Transitional to squamous in the urinary bladder due to urinary bladder stones. Then in cervix, glandular to squamous again. So squamous metaplasia is the most common kind of metaplasia. Thank you guys.